I'm Alison Robertson, a professor and extension field crops pathologist at Iowa State University. And in this episode of Crops TV, I'm going to be talking about some of my research about cover crops before corn, and in particular, that research that pertains to a couple of tactics that you could maybe think about using as you're managing your cereal rye cover crop ahead of corn. The focus of my research, cover crop research, is on winter rye because this is the most common cover crop grown in Iowa. And in a survey that was done by Alejandro Plastino and Jay Arbuckle, 81% of the cover crop in Iowa from 2015 to 2020 was cereal rye or a similar small grain, triticale, for example. And the reason why we grow winter rye here in Iowa is because it establishes well in the fall. It has very good growth in the spring, and it's winter hardy, so it survives these terrible winters that we have. And we're growing it to absorb excess nitrogen, to help with soil erosion, and for soil health benefits. So my research has focused on winter rye because winter rye provides a green bridge for corn seedling pathogens. So what do I mean by that? So in research that was done back in 2016 by Matt Backer, who is working with Tom Casper and I, he was able to show that two soil-borne pathogens of corn, Fusarium species and Pythium species, are able to survive on the roots of winter rye. And what he did was, first of all, he was able to recover those two genera from those winter rye cover crops. And then he infected corn and soybean with those Fusarium and Pythium and got disease on both of those crops. So that's why we say that winter rye is a host for corn seedling pathogens. So the idea here, why we talk about winter rye providing this green bridge, is because what happens when we terminate that rye in the spring, most farmers are using herbicides to terminate that rye. As those rye roots decompose, they release all these pathogens into the soil. And so basically, if we plant our corn into the soil when those pathogen populations are very high, it's like we're planting corn into a petri dish. And so then we can get seedling disease. And so that's what we call the green bridge, is that the pathogens are surviving on the winter rye, and then as that winter rye dies and breaks down, we plant the corn, and then the pathogens are able to jump onto those corn seedlings and cause disease on those corn seedlings. So I'm just going to take a step back and just talk about the traditional principles of plant disease management. And you can see that there are 6 here. And the ones that I'm focusing on in my research are exclusion and avoidance. So what do I mean by that? So exclusion would be and avoidance would be when you're planting when the inoculum is low or not present. So for example, here you can see in this figure below, we have the population of those seedling pathogens, and then we have the days after rye termination. So if we terminate that rye, those populations increase. If there's no corn for that rye to infect, for those populations to infect, then the populations will decrease. And so then we plant the corn when that inoculum is low. There's always a background level of inoculum in fields. And then the other thing we could do is to plant where the inoculum is low or not present. And the best example of this is basically social distancing. So social distancing that corn away from the cereal rye, that can help us manage the seedling disease. And so what I'm going to do in this episode is just talk to you about 2 research projects that I've done where we planted when the inoculum was low or not present. And then we also looked at planting where the inoculum was low or not present. So based on, if we go back to the green bridge idea, we came up with this recommendation, right, to wait at least 10 days to plant the corn after terminating the winter rye cover crop. And that was based on some our first set of research that we did, where we were able to show that if we planted corn within a week of terminating that rye, that we got greater seedling disease. And in 1 year, we got yield loss. In the other year, we got lower yields, but they weren't statistically lower. So the problem with our recommendation, though, as I'm sure that you're sitting there, is that we have these cold, wet springs, right? And so cold, wet springs mean that sometimes we can't get into the field to terminate that rye before we plant that corn. Or if it's cold and wet and we can get into the field, thanks to the cover crop, we can't spray that glyphosate. It's not going to be effective because it's just too cold for it to work. And so as a farmer, that means you either have to wait for it to warm up or the field to dry out and then go in and spray that rye and wait that 10 days and then you plant your corn. Well, you don't want to plant your corn later. You want that corn in the ground as early as possible so that you can maximize that yield potential. Or the other thing you can do is to plant the corn and then to spray the rye that day of planting or up to a few days later. And most of you will know that practice is called planting green. Well, based on our earlier research, that really frightens us. To plant green, that is just, as far as I'm concerned, asking for a disaster. And so we actually set about and set it in an experiment, and we asked, could planting green increase seedling disease and contribute to yield drag? And so the idea here was you have these Pythium populations, right, that are increasing after you plant the rye. And then if those stars on that graph on the x-axis indicate where the glyphosate is sprayed. So if we spray well before planting the corn, so this is where we're going to plant the corn. So if we spray before, 10 days before we've planted that corn, then that population will have decreased and we plant the corn and we're at less risk for seedling disease. If we plant the corn just, within a couple of days of terminating that rye, what'll happen is those populations are going to be increasing as that corn seedling is germinating. And so it's going to be at risk for infection. And then if we wait 6 days and then terminate that cereal rye, those populations are increasing a little bit later during the germination. The question there is the corn plant more resilient then to that infection? And then we have in there that 12 days after planting, terminating the rye, 12 days after planting, and how will that affect seedling disease? So remember that what we're looking at here is the disease triangle, right? So if we have greater populations of pathogen, we have a greater risk of disease. But remember that soil temperature and moisture are also important. What we've found from our early research is that the soil-borne pathogen that's most important following a rye cover crop is Pythium. And so you'll remember with Pythium that's a pathogen that likes cool temperatures, but also wet soils. Some of those Pythium species are producing zoospores that will swim in that wet soil to infect the corn roots. So we got funding from USDA NIFA and set up experiments to look at planting green and how it might affect seedling disease and reduce yield in corn. And so we had experimental plot field trials, and we did those in 2019 and 2020. And then we also worked with some farmers who are part of Practical Farmers of Iowa. And this enabled us to do on-farm research. And so take our experimental plot field trials out onto farms and have a look and see what was going on then. So in the experimental plot field trial, the treatments that we had was to terminate the rye cover crop 18 days before planting the corn, 3 days before planting corn, 6 and 12 days after planting corn, and then we always have a no cover crop control in our experiments. And the small plot trial was on a corn-soybean rotation. The corn was planted no-till. We drilled our rye in late September 2019, and you can see from this picture that we had very good establishment of that rye cover crop and some really nice-looking biomass in spring. And from a nitrogen point of view, we applied 50 pounds of UAN at planting, and then we came back at around about growth stage V4, which was a little bit after we'd sampled those seedlings for seedling disease, and we side dressed with 130 pounds of nitrogen. So we collected a lot of data from this trial, but in the interest of time, I'm just going to stick with seedling disease and yield. But here you can see the year of the treatment, 2019 and 2020. You can see the treatments that we had. And then what I'm showing here is the disease severity that we had on the seedling root. So we looked at the severity on the radicle, and then we looked at the severity on the seminal roots as well. There was no rot on the mesocotyl of the seedlings that we sampled. And so what you'll see in 2019 is that we had the most radical severity there, no difference in seminal severity. But also just note there that we have more radical disease whenever rye is planted. But at 12 days after planting, that's when we had the most disease. And then in 2020, similarly, we saw a lot of disease after planting, significantly more disease than the no rye control. But that 12 days after planting and also that 3 days after planting was a lot more disease compared to the other treatments that we had. So what we were seeing was more severe disease when we planted green, and that's what we were afraid of. We were afraid that planting that corn and then terminating that rye afterwards, that corn would be germinating and starting to grow when there were a lot of pythium around, and those pythiums would be attracted to those exudates from that germinating seedling and therefore attracted to that corn seedling, and there would be a greater risk of infection. What about the yields? So this is 2019 and 2020, and you can see in both years we had a significant effect of treatment on yield. So in both of the years, the yields were lower after we planted with corn following rye, and you'll notice too that the later we terminated the rye, the more the yield was reduced. Okay, so what about the on-farm trials? So in these pictures, this picture on the left here is one of the practical farmers of Iowa that we worked with, and Jyotsna Acharya, who's done a lot of the research that I'm showing you today. And then in the picture on the right, I have Jyotsna again in the bright green pants. And then Sarah Kurtz was a master's student who worked with me, and I'm going to be showing you her research in a couple of slides. And then the young girls on the right are some undergraduates who helped. And this is us out in the field collecting data from the farmers' fields. What did we find here? So in your field notes, you can read up about these trials that we did. I have the links in those field notes, but if we just look at the data here, this is from 2020 and 2021. So you'll see the bushels, the yield that we had. So in both years, we didn't see a lot of seedling disease. So that's why I'm just showing you the yield data. You'll see the farmers' names at the bottom, and then you'll also see the treatments that they had. So they varied a bit from farm to farm, as you might imagine. And at only one farm did we see an effect of planting green on yield, and that happened to be the same farmer in both years. What was interesting about that farm was that there was a lot of biomass in the spring, much, much more biomass than the other farmers who we visited. And we've been thinking about this whole biomass thing. And one of my collaborators who I work with a lot is Stefan Galens at the Practical Farmers of Iowa. And he and Mark Licht and I have really been wondering about cereal rye biomass and what effect it might have on corn yield. And we really think there's a good correlation there. And so Stefan wrote this blog earlier on in 22, April, I believe, 22. And it was very interesting because what he did was, first of all, he correlated cereal rye biomass from the Practical Farmer of Iowa's trials with corn yield and found there was a correlation. But he found that there was a better correlation when he took cereal rye heat units. And so you can see that in this graph. So this is heat units that are accumulated from January the 1st until corn planting date. And the base there is 32 degrees, and that's because cereal rye will start growing at 32 degrees, as opposed to corn, where when we're doing growing degree days or heat units accumulated, we usually use a base temperature of 50. But you can see here that there's a very good relationship, and it kind of suggests that once we get to 800 heat units that have accumulated from January the 1st, it's probably a good idea to terminate that cereal rye to prevent too much biomass from accumulating and therefore reducing yield. But that's a very interesting blog that you might want to look up and have a read. So from my planting green experiment was done just here in Iowa, but recently I became part of this national grant that is also looking at planting green. And this project is called Precision Sustainable Agriculture. And I am leading Common Experiment 2, where we're looking at the effect of cover crop termination timing on slugs, pests, insect pests and beneficial insects, diseases, weeds, and also corn performance. And so we just finished up the second year of the trial. So the first year of the trial was in 2021, and we have a third year of the trial next year. So this slide just shows you the states that are taking part in this trial. So there are 15 states. And I lead Common Experiment 2 and obviously the seedling disease, but John Tucker from Pennsylvania is leading the insect side of the experiment, and Carlene Chase from Florida is leading the weed part of the experiment. And the whole project is led by Stephen Mirsky at USDA ARS and Chris Rehberg-Horton at North Carolina State University. But I wanted to show you some data from the first year of the trial, particularly seedling disease. And so this is the average root rot severity across the locations. And you can see that we have 4 treatments here. So we have a no rye control, and then we have a treatment where we terminate the rye at least 14 days before planting. We terminate the rye 3 to 5 days before planting. So that's where we expect to see a lot of disease. And then we terminate the rye at or after planting. So that would be our planting green treatment. And you can see here the range in disease across those 15 states, and then the averages there. And so we found a treatment effect of at 13 of the 16 sites where we did this experiment. And what we found was the root rot was most severe in the planting green treatment. And so that was really exciting for me as a plant pathologist. Sorry, not if you're a farmer, but for me, because it's not just Iowa that has this problem with seedling disease, that there's obviously something going on with the system. And if we can understand the system better, then we can come up with ways to manage it better so that we can mitigate this yield drag that's occurring. We also collect data on stalk rot, and we found less stalk rot in the planting green treatment. And I suspect that's because those plants tended to be slimmer. The stalks weren't as big and the ears weren't as big. And so there was probably less stress being put on that stalk, on that system. And so there was less chance of those stalk crop pathogens kind of taking over at the end. And then in terms of yield, at 5 of the 13 locations, the yield of the planting green was less than the no rye treatment. And at 2 of the 13 locations, the yield of the planting green treatment was actually greater than our no rye check. Just to summarize this planting green research that I've been talking about, planting corn within 7 days of terminating rye may affect corn growth. So what we saw in the planting green treatment is that the corn seedlings tended to be taller, but they were behind in growth stage compared to the other treatments, probably because emergence was delayed. And then also they have to grow through that cover crop biomass. And so they tended to be very etiolated. We also found that planting green increased the risk of seedling disease. And as I mentioned, that's because rye is acting as that green bridge. But there's probably also some allelopathy and perhaps some nitrogen immobilization going on that's playing a role in this increased seedling disease. And then planting green could result in lower yields. So my first tactic would be to avoid planting green. If you're new to cover crops, one of the things that you want to do is try and terminate that cover crop at least 10 days before you plant that corn. Or if you have very low biomass, then that could also just reduce your risk of disease occurring. Okay, so the next thing that I want to talk about is social distancing corn from a rye cover crop. And so there's a cartoon here showing the corn seed planted apart from the rye, and those purple dots there represent the pythium. So you can see much more pythium around those rye roots compared to that corn seedling. And so if we were to look at this on a graph of the population, we would expect to see lower populations of pythium where the corn seedlings are planted. And then where the rye is planted, we have these high populations. And so as we think about all the technology that we have nowadays, it's not hard to think about precision planting, right? We have all these GPS units, and so that was where we were coming from this experiment, that we should be able to precision plant the cover crop apart from the corn, so that we could then test this theory that social distancing the corn from the rye would reduce the amount of disease. So in the publication that Alejandro and Jay just published, seeding methods, 56% of the cover crops were drilled during that period, 2015 to 2020, followed by aerial and broadcasting those cover crops after harvest. So in 2019 and 2020, with funding from the Iowa Nutrient Research Center, and then Sarah Kurtz, a master's student with me, also got funding from the North Central Sustainable Agricultural Research and Education Center, we established these trials to look at social distancing the corn from the rye. And so what did we do? Well, first of all, we have our no rye control. And then the next thing we did was we planted the rye in between the corn rows. And so the way we achieved this was that we had a 7.5-inch drill, and we plugged 3 of the drills and then just planted 1 row of rye every 30 inches. And then came back in the spring, and we were able to plant the corn down the center. So this meant that the corn was 15 inches away from the rye. And then with the drill again, we plugged 1 of the drills and then planted 3 rows of cereal rye in between where the 2 corn rows would be planted. So that meant that the corn would be 7.5 inches from the rye. And then for our other control, we broadcast the rye, which is what's normally done. And so that meant that the corn would be 0 inches away from the rye. So what did we find in terms of seedling disease? So we're looking at percent radical rot here. And there's some pictures of the seedlings there, so you can see in the numbers of the seedling disease. So this was in 2020. We got some pretty good disease developing. 2019, which was the first year of this trial, we did not see a lot of disease. And what you can clearly see here is that where we planted into the broadcast rye, or when the rye was just planted 7.5 inches away from that corn, we had much greater seedling disease than when the rye was just planted on those 30-inch centers. And the amount of disease we saw was equivalent to the amount of disease that we got in the no rye check. And so this was exciting because it shows that social distancing corn from cereal rye can mitigate that disease. If we look at barren plants, so barren plants are, we always go in and we count the number of plants just before harvest that do not have a harvestable ear. And so those plants would be barren. And the data we have there, the light blue is from 2019 and the dark blue is from 2020. So you can see in 2019, we didn't see a difference in barren plants. We didn't have any seedling disease. But then in 2020, we see a lot more barren plants in the broadcast treatment compared to where we've socially distanced the corn away from the rye. And if we look at the yields reflect pretty closely what we saw in the barren plants. So in 2019, the yields where we planted the cereal rye in 30-inch rows were equivalent to where we had no cover crop. And then in 2020, the yields were slightly lower, but were better than where we planted that corn into the broadcast rye, or when that rye was 7.5 inches away from the corn. We also did an on-farm study. In this case, we were working with the Iowa Soybean Association on-farm network. And so we worked with one farmer there in 2019 and 2020. And what he did was he planted rye in 15-inch rows or in 30-inch rows. And then he also had a no rye strip. And so we were able to go into those strips and compare what was going on there. And we didn't see any difference in shoot dry weight at, would be at V3, V4. There's the growth stage there. So around about V3 in 2019 and a little bit earlier than V3 in 2020. But you can see with the radical rot that we did see some difference. So where we planted in the 30-inch rye, the corn that would have then been 15 inches away from that rye had less disease compared to the corn that was only planted 7.5 inches away from the rye. So that would have been the 15-inch rye. But we didn't see a difference in barren plants or in yield. So tactic 2 for mitigating this yield drag that occasionally occurs with corn planted after cereal rye would be to socially distance the corn from the cereal rye. And so I showed you our work where we precision planted that rye. So we used the drill and planted the rye in different areas so that we could then plant the corn where there'd be no rye. But another way that you could socially distance the corn from the cereal rye would be to do some spring strip tilling. And Mark Licht and I have a student at the Kanawha Research Farm who has done an experiment there. We're just waiting for his data. And then I know I visited a farmer earlier on this year who strip tills and then plants into a cereal rye and has been very successful doing it that way. So we're following up on this research because there are some potential shortfalls with precision planting, right? Or just potential shortfalls with planting the rye in different arrangements. And probably the most obvious one is that where we're planting just 3 rows of rye in between the corn, that means we have 25% less cover on the landscape. And when we're planting that rye just in 30-inch rows, then we have 75% less cover on the landscape. And so the obvious questions there are, what does that mean for for soil erosion, for water infiltration? What does that mean for soil health if we've got so much less rye on the surface of the landscape? And what does that mean for nutrient cycling? So we recently got funded from Iowa Nutrient Research Center to start looking at some of this coming year. So in summary, tactic 1 would be to plant, if not to plant green, because planting into a green rye cover crop can negatively affect corn. It delays emergence, there's increased seedling disease, and that can lead to a yield drag. And then tactic 2 would be that some sort of precision planting, so plugging the drills or maybe using spring strip till could be advantageous for corn, because we see better corn growth when that corn is socially distanced from that rye. We see reduced corn seedling disease, and because of that can mitigate that yield drag. Lastly, to finish off, is seedling disease only to blame? And this is kind of some other questions that we have here. So I love this quote from a farmer in Montreal: corn is a princess plant. And I couldn't agree more. Corn is really finicky, and we really have to look after it. And as opposed to soybean, which is so much more resilient, and we just don't see any of these problems with soybean following a cereal rye cover crop. But some of the things that I think are also playing a role in this system are, with all that cover crop biomass there, we do have cooler and wetter conditions that corn plant is germinating and emerging in, so not the best conditions. We've been doing some work on allelopathy in my lab, and Jyotsna has been able to show that when we have allelochemicals present as well as pythium, that we get more severe seedling disease. And then right now, Mark Licht, Marshall McDaniel, Peter O'Brien, and I have funding from the Iowa Nutrient Research Center to look at nitrogen immobilization and a cereal rye cover crop, and to try and understand how nitrogen immobilization might be affecting seedling disease development. So I want to thank you for supporting Crops TV. And if you have any questions or comments about this presentation, I'd be very happy to take them, and you're welcome to email me. There's my email address right there on the screen. And I also have a Twitter account if you want to tweet me. So thank you for your attention, and enjoy the rest of your day.